Okay guys, we're going to start the test 4 review. This is going to go over meiosis and genetics, so let's take a look at a few of the topics. The first main topic, guys, is you need to make sure that you can understand the difference between mitosis and meiosis. This is going to be mostly kind of like a matching type question, or it may ask you, give you like characteristics, and it'll ask you which undergoes those particular steps, which would be mitosis or meiosis. So if you look here, this is straight out of your notes. You'll notice they have mitosis, that's associated with asexual reproduction. Remember that it produces 2 genetically identical daughter cells. This is to help give us genetic stability. Remember that stem cells do this process, also known as somatic or body cells. They do 1 nuclear division and it produces 2 cells because you divide from 1 cell to 2. Meiosis, on the other hand, is associated with sexual reproduction. It produces haploid daughter cells, meaning they're going to half the number of chromosomes. This leads to genetic variability. Now, the only cells in the body that do this are the germ cells, and the germ cells are found in the gonads. So in females, they're going to be found in the ovaries, and in males, you're going to see this in the testes. Now, remember that they go through 2 cell divisions, and since they have 2 cell divisions, that means we ultimately end up with 4 cells. Now, in the chart, it shows you there is an exception for oogenesis, but we're just going to remove that exception and just say yes, it forms 4 cells. Now, it's also important for you to be able to ID which stage the cell is in, whether it's mitosis or meiosis. Now, we're going to focus mostly on meiosis since that's the chapter we're looking at. So if we're looking at this particular picture, take a second and kind of look, what phase is this in? Now, it should be relatively easy to say, oh, well, that's anaphase. But remember that in meiosis, it can be anaphase 1 or anaphase 2. A key characteristic is that those tetrads are being pulled apart. Tetrads are only seen in meiosis 1. So this right here would be a picture of anaphase 1. Also, another key indication is that there's only 1 cell doing this. If it was anaphase 2, there would be 2 cells undergoing this process. So if we look here, this one has the tetrads present, crossing over is taking place. However, the nucleus hasn't completely disappeared, so which phase is this? Well, it would be prophase 1. All right, this one we see the cell is almost completely split. The nucleus is reforming, so this should tell you that it is telophase. And since we're going from 1 cell to 2, this is telophase 1. All right, the last phase for meiosis 1 would be metaphase. If you notice here, the tetrads are lined up in the middle, so this tells you that this is metaphase 1. Now, do you see a distinct difference here? Instead of having 1 cell dividing into 2, I now have 2 cells dividing into 4. This one is showing you that this division is almost complete, so this would be telophase 2. Here, it's getting ready to divide. We see that it has the nucleus is starting to fragment. We can see that there are 2 cells going through prophase, so this is prophase 2. Again here, chromosomes are lining up in the middle. This is metaphase 2. And so hopefully that helps you review and be able to identify. Now, I left out anaphase 2, but anaphase 2 would just be 2 cells going through the process of the chromosomes being pulled apart, and that was what you would see with anaphase 2. So now let's move on to some specific type questions. In your review, it talks about meiosis 2 is most like what other process? Well, guys, if you look at the picture that you see here, you'll notice that this looks very similar because this is meiosis 2. Meiosis 2 looks very similar to mitosis. It looks very similar to mitosis. The next question, know how to find the diploid and haploid number of chromosomes for an organism, and more particularly man, but it doesn't matter what type of organism we're looking at. Remember that the diploid number is 2n, okay? So it's when you have 2 sets of chromosomes. The haploid number is n. So in humans, the diploid or 2n number is 46, which means the haploid number would be 23. Now there's another way we could say this. On the 46 side, we could say that we have 2 pairs of 23. The pairs tell you that there are 2 times 23, which is 46. Okay, so make sure that you pay attention to the wording, because if there's pairs, that means there's 2 sets. Now, the purpose of meiosis and fertilization. Well, let's look first at meiosis. Meiosis is the process that produces gametes. The whole process of meiosis is that we want to half the number of chromosomes to create haploid sex cells. So these cells are going to have N chromosomes. On the other hand, fertilization is the opposite. Fertilization is where we're going to fuse an egg and sperm together to restore the diploid number. Now, what's unique about this is since we're restoring the diploid number, we are going to have a unique individual because this is what helps us create genetic variability. So, how does genetic variation occur in the process of sexual reproduction? Well, we already just talked about one, which was fertilization. Fertilization allows for this random egg and sperm to come together to give us a unique set. But also remember that crossing over can take place. Remember that in crossing over, a piece of one chromosome breaks off and switches places with another. Now, these are the homologous chromosomes, so meaning it's a piece of one switching places with piece 2. So it's the exact same chromosome, just giving more variation. Now remember also independent assortment. Independent assortment just means that chromosome 2 doesn't care what chromosome 5 is doing. Chromosome 5 doesn't care what chromosome 8 is doing. They're going to independently assort into different cells on their own, independently. Now, during which stage of meiosis does the homologs or tetrads separate? Okay, so when we look at this, homologs or tetrads meaning we have a set of 4 chromosomes. And if they're separating, we know that this is anaphase. So the tetrads are only found in meiosis 1, so this would be anaphase 1. The next question says, when does interkinesis occur? Interkinesis is very similar to interphase. However, it is missing the S phase with DNA replication. So interkinesis is going to occur between meiosis 1 and meiosis 2. Since there's no S phase and no copying of the DNA, we can't call it interphase, but we call it interkinesis. It connects meiosis 1 to meiosis 2. Now, what is the purpose of crossing over? We just discussed that. Genetic variability, okay? But when does it occur? Well, it has to be when those tetrads form. The tetrads form in meiosis 1, and the crossing over is allowed to take place during prophase 1, okay? During prophase 1, all right? So this is going over some specific type questions. Now, there is going to be a matching with a picture that you're going to need to know the diplonic life cycle of a human. Now, the picture in your book is a little bit better, but here we have a male and female. They both can undergo meiosis, which is spermatogenesis in male to create sperm, and oogenesis in female to create egg. Now we've half the number of chromosomes. When this egg and sperm come together, this is fertilization. This creates a zygote. This zygote, this new cell that's going to be a new individual, then undergoes mitosis. Once it undergoes mitosis, it goes from 1 cell to 2, 2 to 4, and so on until we get a new individual. This individual will continue to go through mitosis until, of course, they reach adulthood. Now, during puberty, we will see that mitosis still continues on in the body cells, but when puberty hits, now the egg and the sperm can start to be formed in the ovaries or the testes, and the cycle then can continue. Now, there's a number of things that could be asked here. It could remove spermatogenesis, and there could be a letter A there, and it says, what process is this? Well, if it's a male creating sperm, obviously that is spermatogenesis. It could take out the word fertilization, and it asks you what this is when an egg and sperm fuse. Well, that would be fertilization. Okay, so study this particular picture, and this is in your notes as well. All right, so that kind of concludes the meiosis and sexual reproduction side. So now let's talk a little bit about the genetics. So genetics is the study of what? Well, genetics is the study of heredity. Heredity are the genes that can be passed on from generation to generation. Now, this brings us to some terms we need to know. We need to know about codominance. Codominance is where both alleles are expressed. One is not dominant over the other. And one of the best examples we've talked about in class is AB blood type. If you're AB, you have A protein and you have B protein. You have both. So they're dominant. It's AB blood. We also see that we need to know what sex-linked disorders are. Sex-linked disorders are disorders located on the sex chromosome, meaning the X. Okay, remember we don't focus on the Y because the Ys are only found in males, so we're not going to focus in on that. It affects males more than females, however, since females have an extra X, because remember, females are XX, whereas males are XY. An example of this would be red-green color blindness. However, we did discuss several others like hemophilia, muscular dystrophy. Those would also be examples of X-linked disorders. Now remember that our genetics can be influenced by environmental factors. These factors that influence how genetics are expressed would be things like temperature, diet, drugs, all talked about within your notes. Now, codominance we said was where both are seen equally, A and B blood type. However, in incomplete dominance, one allele is not completely hidden by the other. So you see an intermediate. This was best seen when we saw the 4 o'clock flowers or carnations, where a red flower is mated with a white flower and it gives you the pink. So the heterozygous individual who has the red and the white gene, the flower is therefore pink instead of red or white. That's incomplete dominance. Pleiotropy. Pleiotropy, remember, is one gene affecting many traits. Okay, remember this is where we saw that collagen could be affected. That's one protein, and it causes Marfan syndrome. Or hemoglobin is defective, and hemoglobin is one protein again, but it can cause sickle cell disease. Now, non-disjunction, guys. Non-disjunctions kind of go hand in hand with what we saw with meiosis. This occurs normally during meiosis 1 or 2, and it causes an abnormal number of the chromosomes. So remember when the chromosomes are supposed to go in anaphase and they're supposed to pull apart like this? In non-disjunction, they don't. The whole X goes in one direction, leaving the cell over here having one chromosome less, and the cell over here having one chromosome more. So that's what we're talking about with non-disjunction. Now, polygenic inheritance. Polygenic inheritance are where many genes are affected by one trait. Okay, so we saw with pleiotropy, it was one gene affecting many traits. Well, here, guys, it's many genes affecting one trait. Okay, so like height or skin color. Okay, there's a lot of intermediates, and it depends on how many of the dominant genes you have versus the recessive, and it's many genes working together. All right, so the next bullet says be able to determine the genotypes of parents when given information about their offspring, whether it's dominant or recessive. So a little key thing here, remember that the dominant trait is the capital letter. Okay, it's represented by a big letter, so in this case, big T. Now, both big T, and big T, little t will show the dominant trait. Okay. Recessive, however, is the little letter. It's the one that is hidden if the dominant is present. You will only see this if there are 2 little letters, or in this case, 2 little t's. Also know, when you do a test cross, you need to use this to determine if the parent with the dominant phenotype is a big T or a big T little t. And remember, we talked about this in the notes where you did 2 separate Punnett squares. If all of the offspring end up being tall, then you know that that parent was big T. However, if half of them come out short, then you know that that individual was big T little t. So what does a gene's locus refer to? That is the gene's location. Locus kind of sounds like location, so it's where the gene is located. You also need to know Mendel's law of segregation and independent assortment. Now, these came straight out of your notes. Remember, the law of segregation is where each individual contains 2 alleles for each trait. These alleles separate during meiosis, so each gamete contains 1 allele. Fertilization restores the 2 alleles in an individual. So segregation, guys, means that they split, they separate. So this way, there's 1 chromosome 1 in one cell and 1 chromosome 1 in the other, and this came from the mom. You then get your 2 copies of chromosome 1 again when the egg and the sperm fuse together. Remember, the law of independent assortment says that each chromosome separates on its own. Remember, chromosome 2 doesn't care what chromosome 5 is doing. They separate on their own. Okay, now, know how and when to use the addition rule and the multiplication rule of probability. Now, I told you these 2 kind of concepts. With the addition rule, you're going to add them together. This is a this or that type of scenario. This or that. So they're looking at, okay, what are the chances that it might be this or this other trait? Now, when you do this, you're going to add them together. Now, remember, these are fractions. So if I did 1/4 plus 1/4, that equals 2/4 or 1/2. Okay. The multiplication rule is a little different. The multiplication rule is this and this, meaning what are the chances that both of these things happen? Okay, in 2 separate individuals. This means you have to multiply. So let's just say that there's 1/4 of a chance for the first one and 1/4 of a chance for the second one. Well, I want to see what's going to happen if they both end up that way. Well, that's 1/4 times 1/4, which equals 1/16, because 1 times 1 is 1 and 4 times 4 is 16. So this is just to review the addition rule and the multiplication rule. Now, what are test cross used for? Test cross, remember I discussed on the last slide, are used to determine if a parent with a dominant phenotype is homozygous, big T, or if they're heterozygous, big T, little t. What are karyotypes? Well, guys, karyotypes, remember, is important in diagnosing certain diseases. And karyotypes are where the homologous chromosomes are paired together, forming a picture or pattern. Okay, autosomes and sex chromosomes are classified separately, and this allows us to see if there's extra copies or if there's not enough. So something like Down syndrome, if we saw 3 copies of 21, chromosome 21, that lets you know that that child would be diagnosed with Down syndrome. Okay, so it's used to diagnose trisomies or monosomies, like in Turner's where they only have 1 X. Okay, know how to read and use a pedigree chart. Now remember, in a pedigree chart that females are represented by circles, males are represented by squares. Colored in shapes are affected. So if it's a colored in square, it's affected male. If it's colored in circle, it's an affected female. A line between a circle and a square represents mating. And there are 3 types of pedigrees. Pedigrees can be autosomal dominant, autosomal recessive, and X-linked recessive. Now remember, in autosomal dominant, you're going to see lots of shapes colored in. But you're going to see a relatively similar amount of male and females. In autosomal recessive, you're not going to see very many shapes colored in, and you're going to see still a relatively similar amount of males and females. In X-linked recessive, still we're going to only have a few colored in, but we're going to see that most of the shapes colored in are squares because these disease affects males more often. So if we were to look at this particular example, okay, there are 3 shapes colored in, but the 3 shapes are squares. This should tell you that this is an X-linked recessive disorder. On the other hand, with this pedigree, you'll notice that there are a lot of shapes colored in. And there are 1, 2, 3, 4, 5 males, 1, 2, 3, 4, 5 females. See, relatively the same. Now, since they're the same, this would be considered an autosomal dominant. Okay, so review these in the book if you need some help. Remember, though, that the paper on the pedigree charts is due with your portfolio. All right, what determines the gender of an individual? Well, the gender of the individual, guys, is determined by the sex chromosomes. The sex chromosomes, if it's an XX, it's a female. If it's an XY, it's a male. Now, remember, the Y is maleness. So if the individual has just 1 X, they would be considered female. Know the basics of the following disorders. Now, these are given in the disorders presentation that we didn't necessarily go over each and every one of these, but you do need to review them and know them. Cystic fibrosis. This is an abnormally thick mucus secretion. It's most common lethal genetic disease among Caucasians. This is a recessive disorder. If you get the 2 little letters, little c, you can have cystic fibrosis. It makes your mucus extra thick, causing you to have other increased infections like respiratory infections and digestive problems. Down syndrome, remember, is a trisomy, and it's a trisomy 21, meaning you have 3 copies of chromosome 21. Hemophilia. Hemophilia is the inability to produce normal protein clotting factors in the blood. So this individual could bleed to death from a relatively simple cut or bruise. Klinefelter syndrome. Klinefelter syndrome is also a trisomy like in Down syndrome. However, it's an XXY. This individual is a male, but this male will have underdeveloped testes, breast development, and subnormal intelligence. Now, when I say subnormal intelligence, this just might mean that they have like a learning disability. They're dyslexic. They don't get math that well, but there's a lot of us that could have difficulties like this, and we don't have Klinefelter syndrome. Sickle cell disease. This is a disease due to the production of defective hemoglobin. This makes the red blood cells look like bananas or crescent moons. Now, because of this, it causes poor circulation and it makes the patient anemic. Last on this particular list is Tay-Sachs. Tay-Sachs is a disease most prevalent among Jewish people, and it's inherited as autosomal recessive disorder. Now remember that this is a defective lysosome. The lysosome organelle is defective, and remember, they're the ones that recycle and clean up the cell. So this can cause a major issue, leading to death by the age of 4. All right, so now let's look at some terms, guys. With terms, you need to know what an autosome is. Autosomes are chromosomes containing genes that determine traits other than sex. So these are the non-sex chromosomes. Diploid means you have a full set of chromosomes, 2N. Remember, the cells that have 2N are your body or somatic cells. Haploid is the number of chromosomes denoted by an N found in gametes, meaning you have half the number of chromosomes. Heterozygous. Heterozygous is a big letter, little letter. They're different. This is also termed a hybrid. Now remember though, in Mendel's experiments, Mendel's first generation, the one right after the parents, were almost always hybrids. Okay. Homozygous also means same, so these are purebred, the genotype that contains two identical traits, so they could be homozygous dominant, big T, or homozygous recessive, little t, little t. We also see genotype. Remember, genotype is the genetic makeup of an individual, whereas phenotype is their physical appearance, which you're actually seeing. A gene is the unit of heredity, whereas the allele is an alternative form or variation of the gene. Remember that we represent genes by like big A, or big A, little a. Alternative variations, or the alleles, would be the capital A, which is the dominant allele, and the little a, which is the recessive allele. Okay, well, this brings us to dominant or recessive. The dominant is allele that is expressed in the heterozygote. Okay, it's the big letter. It's the dominant letter. A recessive allele is amassed in the heterozygote, so it's the little t. You don't see it, but they're carrying it. So you might see that trait later on in future generations. Now, this one says you need to know what to expect when we talk about ratios from a monohybrid or dihybrid cross. So take a second here. If big T is tall and little t is short, you are crossing a heterozygous tall with another heterozygous tall. Well, what would you expect? Well, you just do the Punnett square. When you do the Punnett square, you'll notice you get 75% tall and 25% short. If you did this as a ratio, this would be a 3-to-1 ratio because this is a monohybrid cross. All right, what about this one? Big T crossed with little t, little t. If you were to do the Punnett square, you would find that you would have 100% tall. Every box would have a big T and every box would have a little t. But remember, since the big T is dominant, they're 100% tall. Okay, what about this one? Big T, little t crossed with two little t's. Again, if you do the Punnett square, you can pause the video and do the Punnett square. You'll find, though, that you get 50% tall and 50% short. So make sure that you can understand how to set up and do the Punnett squares to find the ratios or the percentages. Now it gets a little harder, guys. These are the dihybrids. The dihybrids is, if we see here, we're still talking about tall, but now we're going to add in green, big G, with a little g, which is yellow. So in this particular case, I have a heterozygous tall green plant crossed with a short yellow plant. Now, if you were to do this particular Punnett square, okay, you would end up getting 4 tall and green, 4 tall and yellow, 4 short and green, and 4 short and yellow. This is very similar to the one that you saw in your notes. We talked about the fly's wing length and their color. Now, what about this one where you cross 2 heterozygotes? This one is very similar to one of the examples in your genetics problems. This one gives you a weird ratio. This is 9 tall and green, 3 tall and yellow, 3 short and green, and 1 short and yellow. So this gives you a unique 9-to-3 to 3-to-1 ratio. All right. Now guys, autosome chromosomes in somatic cells versus germ cells. In somatic or body cells, remember you have 44 autosomes or 22 pairs. You also have 2 sex chromosomes or 1 pair. Now this equals your 46 total chromosomes, or if we were looking at pairs, 23 pairs. Now in germ cells or the gamete, it's a little different. You have 22 autosomes, 1 sex chromosome, and that equals your 23 chromosomes. There are no pairs here because we've already removed the pairs so that the egg and the sperm, when they fuse, that returns that diploid number. All right, classification of chromosomal mutations. Now guys, remember polyploid means that the cells have more than 2 chromosomes per set. This is only seen in plants where they get 3N or 4N. They just get a whole new complete set of chromosomes. Instead of 2N, they have 3 or 4. Monosomes is a loss of a complete chromosome. This is 2N minus 1. A trisomy is an addition of a complete chromosome, so this is 2n plus 1. So guys, the monosomy for a human, or when we talk about a human, is 46 minus 1, which would be 45. A trisomy would be 46 plus 1, which would be 47. Now remember guys, a deletion is a loss of a portion of a chromosome. Inversion is where you take a piece of it, you break it off, and you turn it upside down. It gets turned 180 degrees upside down and rejoined. A duplication is where you take a piece of the gene and you duplicate it. So it would say A, B, C, D normally, but now it says A, B, C, B, C, D. The B and C were duplicated. Translocation, this is where we take and break a piece off and it moves to a completely different chromosome. So a piece of 2 breaks off and it gets added on to chromosome 11. So that's translocation. All right, last but not least, when we're talking about these particular disorders, you need to be able to tell if a genetic disorder is autosomal dominant, autosomal recessive, or X-linked recessive. Now all of these are either been talked about in class or they have been presented in that PowerPoint over the genetic disorders. So Huntington's disease was discussed in class. Huntington's disease is a dominant disorder, therefore it's an autosomal dominant. Hemophilia was discussed, and that affects males more, so that's an X-linked recessive. Cystic fibrosis, earlier in the PowerPoint I also gave you this answer. It's autosomal recessive. Phenylketonuria or urea, this one is PKU. It is also an autosomal recessive. Duchenne's muscular dystrophy is an X-linked. Muscular dystrophy is a sex-linked. It affects males more than females. And neurofibromatosis is an autosomal dominant. So if you look here, you have 2 dominants, 2 autosomal recessives, and 2 X-linked. Guys, these are going to be kind of like a matching. So make sure that you know what each of these are. And if you need to, look them up in your notes. All right, so I hope this PowerPoint helped you review for your exam. If you have any other questions, please feel free to email me and be ready to take your exam coming up on Monday afternoon in lab.